All right, kidney development. Moving on, we've talked in the last few sections about the GI tract and the respiratory tubes and liver and stuff like that. So let's finish up with some of these internal organs. We're going to talk about kidney and the heart. So the kidney development. Let's get my pointer once again, more so I can see. So remember, we have our neural tube and our notochord kind of front and center here. Then we have the paraxial mesoderm, which turns into the somites, which you get the head, somites, sclerotome, dermatome, myotome, syndotome, etc. We talked about that development. And then moving further out, we have the intermediate mesoderm. And so that's where the kidneys and some of the gonads. Remember, the germ cells are preserved very early on from conception, from fertilization. But we have the rest of the gonad tissue, right? And that makes sense that it develops with the kidneys, right? Because of the similar path, especially for males sharing the urethra for both reproduction and for kidney waste disposal, etc. All right. So let's keep going. And so here we're just kind of identifying the medial mesoderm compartments. This is a 12 somite chick embryo. You can see the somites here. So it's about 33 hours. So there was labeling here for cordon, which is a notochord specific mRNA, which is blue. And then we have paraxis, which is a green mRNA, which is in the somites. You can see these little balls like we talked about, the somite development along the side of the notochord there. And then we have PAX2, which is in the intermediate mesoderm, which is the red. Okay, so this is going to be a lot of information on this slide because, whoops, oh, that's not what I wanted to do. There we go. Okay, so this is a general schematic of the development of a kidney, a vertebrate kidney specifically. So the nephrogenic cord, which is this kind of pink-red region here, it can also be called, it's from the intermediate mesoderm. It develops from that, so sometimes it's just called the intermediate mesoderm or the nephrogenic mesoderm. But it's a critical embryonic structure that gives rise to the urogenital system. So your gonads, like your whole connected gonads and bladder and all that stuff, all arises from this nephrogenic cord. So some of the kind of features of this, so it develops from that intermediate mesoderm. Obviously, we talked about how that's kind of what we're looking at this unit. And this is located between the paraxial, so the one real close to the notochord neural tube. So it's located intermediately between that and the lateral plate mesoderm. And it extends longitudinally along both sides, so up the backbone or up the notochord. And this begins to form during the third week of human embryonic development. So again, this is this extending or growing nephrogenic cord serves as precursor to some of this early kidney development. So the kidney development, there's kind of like three systems or stages that develop sequentially. So we have this pronephros up here, which is like the first to kind of get created by the developing or the moving and growing nephrogenic cord. The pronephros is the first very primitive kidney system that is still vestigial, so it still kind of exists in humans, but it's laid down early in development and is used early in development, but it is very primitive, so it's not like the final filtration that the kidney does. It's not really involved in that. And then we have the mesonephros, which is these kind of little additional longer fingers here that are laid down. Those are the second intermediate kidney structure, I guess is the best word for it. And it is used very briefly in development until in the period between when this process starts and when the kidney is developed enough to be functional. The mesonephros does some of this filtration. And then finally we have the metanephros. So you see here this metanephrogenic mesenchyme. That is the final kidney system that develops into the permanent adult kidneys. So you can see as it moves down here, we get this ureteric bud that comes off of the nephric duct here. This is the nephric duct. We get this ureteric bud that comes and it connects to the mesenchyme tissue here, or it grows into the, we'll see how the reciprocal induction of this works coming up, I think, in the next lecture. But it grows into this mesenchyme tissue and then starts to form the kidney there. Okay, I think we've covered everything. So the gonad would develop kind of in the proximity up here of where the nephric cord passed by. And did we get everything? I think we covered everything. Okay. So this is just a kind of an image of what's going, like a micrograph of what's actually happening. So this is an intermediate mesoderm at 13 days, showing the induction down here of the kidney. That we're going to talk about this reciprocal induction where we get this metanephrogenic, it's always a mouthful, metanephrogenic mesenchyme here, and then we get this tissue, this uterine bud that grows into it and starts to form all these branches that we see like in a kidney. And so how they stained this, the duct tissue is stained with a fluorescent antibody to a cytokeratin molecule that is found in the ducts and the derivatives of the ducts, and not found in the rest of the tissue. So that's how they labeled it. So we do have some, when we talk about this induction, tissue-specific induction or reciprocal induction, when we get into that next, there is some like juxtacrine and paracrine signaling, right, going on. So signals from the paraxial mesoderm induce this kidney formation in the intermediate mesoderm, and this is in a chick embryo. So you can see we have the paraxial mesoderm, right? So that's kind of where the somites are going to develop. And then we have beyond that the intermediate mesoderm, which we are talking about the induction here. And then we have the lateral plate mesoderm further out. And this is what the lateral plate turns into, like your limb buds and things like that. Pelvis, I think, if I remember right. Anyways, so the intermediate mesoderm here signals or is getting communication from the paraxial mesoderm. So in this experiment here, they just took a needle and just cut in between the two tissues to separate that and see if we'd still get this kidney induction or kidney development to occur. And so after they let, they didn't give me a date of how long this was after severing or cutting in between. So it results in this gap. So you can see here's like the somites here, or like it looks like the somites, those little bumps there on each side. So this would be your neural tube or notochord, depending on what direction we're looking at it from. And then the somites on the right hand side, and then this is the cut that just grew real big. And then you have the mesoderm here, right? So it's kind of the big gap in between. But what you can see is that they can stain here using PAX2, and that is only expressed in certain areas, and so that is expressed in the pronephric kidney, so like the early kidney stage that we talked about earlier. And you can see that if you separate it, it doesn't occur on the side that you separate if you cause a big gap between the two mesoderms, so the paraxial and the intermediate mesoderm. So there's some sort of juxtacrine or paracrine factor signaling there. So, all right, short one. I wanted to, before we get into like this reciprocal induction and stuff going on in the next unit, just kind of give an introduction and make it so it's not a long lecture. So that is it for kidney development. We'll get more into kidney development, part 2, reciprocal induction of the kidney, in the next video. So thanks for watching, and I'll catch you in the next one.